What is a virus? A presentation by Professor Gershom Zajcek, Faculty of Medicine, Hebrew University of Jerusalem. The Gaia hypothesis was formulated by the chemist James Lovelock and co-developed by the microbiologist Lynn Margulis in the 70s. I regard Gaia as a theory of biology, the time dimension of biology. In a previous presentation I discussed part of this project, and here you have my conclusion then. In the beginning of Gaia was a cell. All cells come from cell. Each cell has a self, and provirus stores the self. Virus is a cell organelle, and virus is a messenger of the self. So here you have the young Gaia. It started here with a single cell, then Gaia evolved in cell families, first prokaryotes, then eukaryotes. Later on we have this multicellular life, and currently I discuss the vertebrate state of Gaia, but today we are interested mainly in the first single cell Gaia. So we are talking about the relationship or the formation of viruses in prokaryotes and eukaryotes, and all viruses known today were created during this time. So here's some definition. Virus is a cell organelle formed by the provirus. Provirus is a set of cell genes that control virus creation, cell receptor formation, and stores the self of the cell. Following virus creation, cell dies by lysis and the viruses exit the cell. Virus infects only cells with the same self as that which it carries. Virus infection triggers the formation of provirus. So, first of all, we want to understand why we need a self in each cell. And this can be deduced from the fact that the Gaia is an open system in homeostasis, and you cannot create a homeostasis without the formation of the self. So in order that homeostasis will be maintained, you need a self in each cell as a marker of homeostasis, and only then homeostasis can be maintained. So we start again with the Baltimore classification, which describes the relationship between 7 kinds of virus or 7 classes of viruses and the unicell. So each of these viruses interacts with the cell and creates the formation or triggers the formation of a provirus. Provirus is a set of genes which control the formation of messenger RNA. They store the self, and the messenger RNA is controlled to form viruses, and also there is a messenger RNA for the control of the receptor. So provirus protection is innate immunity. Provirus controls the receptor and blocks infections by viruses carrying a different self. The interaction between virus and cell and the formation of the provirus I call virus-provirus process. It describes the stages in which the cell changes after the formation or trigger or infection of a virus. So we start with a virus, and after attaching to a receptor, the virus is uncoated and only the genome of the virus enters the cytoplasm, and it is manipulated by enzymes until a provirus is controlled, and the provirus then stores the self and controls the formation of virions. And once the virions are formed, the cell dies and they exit into the environment. And what's important to understand that the virions are messenger of the self which was created by the provirus. So they carry in the genome the self which determines what cell they are going also to infect. So here is the same description of this process in a different way. We are talking about the retrovirus, RNA virus, which infects a cell. After attaching to the receptor, the retrovirus is uncoated, and its genome, the naked genome, is manipulated by enzymes of the cytoplasm until a DNA copy of this genome is formed, or I call it DNA image. And once this DNA image is formed, the DNA fuses with the cell DNA, and the provirus is formed and performed whatever I just described. So, conclusion: the origin of viruses is the origin of the cell in which they were created. Virus and cell is one system which is controlled by a provirus. All cells come from the cell, and each cell has a self. Enjoy.